So good morning, everyone. Thank you for joining BrainMap today. I want to remind you that this BrainMap Seminar Series is co-sponsored by the B41 funded Center for Mesoscale Mapping housed in the Martinos Center. It is my pleasure to introduce our today's speaker, Dr. Kristen Kennedy. Dr. Kennedy is an associate professor of psychology in the School of Behavior and Brain Sciences and the head of cognitive neuroscience at the University of Texas. She's also associate professor at advanced imaging research at the Southwestern Medical Center and an affiliate of the Alzheimer's Disease Center there. Dr. Kennedy's research centers around health and lifestyle modifiers of brain aging and their cognitive consequences using multimodal imaging techniques. Today, she will talk about aggregating brain structural and functional properties to better understand cognitive aging. Dr. Kennedy, the virtual stage is all yours.
Thank you. Thank you for the invitation. And thank you especially for attending, because I can empathize with the fact that the Zoom fatigue is very, very real at this point. So thank you for fitting one more talk into your schedule. And thanks for inviting me to let me talk about one of my favorite things, which is understanding what of all of the things out there in the world most predicts our aging of our cognitive systems. And so that's what I'll talk to you about today. So let me get all of these other things off my screen. Okay, so one of the things that it's very apparent when you start looking at trajectories of aging maps across the lifespan like these is that, yes, there is a fairly steep decline of cognitive performance across the lifespan, even in cognitively normal, healthy adults. That's probably no surprise to anyone here. What I think is probably more interesting is that there's a great deal of inter-individual heterogeneity in the individuals in these research studies. And so, for example, you can see that there are individuals in these green circles who are older and who are outperforming many of the other participants in the study. And you could argue that this is kind of our goal, is to fall somewhere above the regression line as we age. This is just an example from working memory composite of several different working memory tasks. And you can see this is about a negative 0.44 slope here, and even stronger slope here for something more higher order cognitive function, like executive functions here. This is a negative 0.58. But still, there are individuals who fall above the performance of the other people in the cohort. Correspondingly, there are individuals who aren't performing as well at older ages, and they can be located down here. And again, you could argue that the goal of aging is to try to fall in the green circle and not the red circle. And so trying to determine or predict of all of the factors out there that have to do with our lifespan, what most determines how we get here or how we get here is a surprisingly complicated and multifaceted question. And indeed, this is the focus of many, many people's program of research to try to figure these things out. And this is, of course, one of the things that I study. So what determines how we end up in this group or this group? And so how we end up in these different cognitive outcomes is really a complicated mix of synergistic influences, like this conceptual framework here illustrates. So age itself as a variable, we know, is just a catch-all umbrella term, really. It's how long we have been existing on this planet. And what it really encompasses is all of the things that have happened to us for good and for bad that we bring with us across our lifespan. And so anytime we're interested in looking at the effects of aging on, say, brain structure or brain function, it's always a product of the modifiers of this association by different genetic predispositions. Again, some neuroprotective, some as risk factors, and our health factors, our lifestyle factors, all of the conditions that we aggregate across a lifespan, the most salient and common of which to the aging process, arguably, is vascular risk. And that's one of the things that we hone in on in our study too. And so then we have the things that moderate the effects of age on brain structure, and then the interplay of our brain structural properties and our brain function properties is then modified by its own sets of factors. And these could go on and on and on. These are deposition of beta amyloid plaques, the occurrence of tau, leaky blood-brain barrier, neuroinflammation. You could drag this list out on and on and on. And so the culmination of these effects on our brain structure and our brain function, and then how our brain structural properties give rise to our brain functional abilities, and probably also slightly reciprocal. But for today, I think we'll stick mainly with the fact that our brain structure is what gives rise to our brain's function. The outcome of all of this totality is what sets each individual on a trajectory either toward pathological aging outcomes such as Alzheimer's disease, or sets us on a trajectory toward a more healthy aging outcome. And in any case, we're very interested in one's cognitive capacity or cognitive ability. But here, when we're talking about healthy aging, we're talking about cognitive performance that's well within the normal range. But as we saw on the last slide, there's definitely heterogeneity in that. And what separates people from kind of that upper quadrant and that lower quadrant is very interesting and important to study, with the ultimate goal of mapping these things and understanding how the brain arises our cognition and our cognitive aging, so that we can use this research to inform clinical practice of ways to help maintain good cognitive function until old age. And so that's kind of the conceptual framework that I'm operating on. And so I also wanted to mention that in any study of aging of the brain and aging of cognition, I think has three important facets that it needs to consider or incorporate into those studies. The first one is that we need to acknowledge and embrace this heterogeneity in different people's aging trajectories. And so we need to think about individual difference factors as much as we can when we think about aging of the brain and aging of cognition. And so our research tries to take into account as many health, risk, protective, lifestyle factors that we can when we test these models. The second is that we know that the different properties of the brain don't age or operate independently in isolation. And so using multimodal neuroimaging approach just to look at the additive or synergistic effects of different types of brain aging on each other, and then ultimately on cognition, I think is the only way forward to really understand how people age cognitively. And then the last one is, I think that any study of brain aging needs to go beyond looking at the effects of age on brain properties. And it's necessary to yoke these aging brain associations to one's cognitive performance as the output and the desired outcome measure. One, this helps the interpretation of some of the findings that we have and some of the directions that these associations and effects can take, especially as we know the brain is highly malleable. And so neuroplasticity can have us interpreting factors in either direction sometimes. And so I think that this is an important way to yoke that to something that is a known entity. We know that people should have better cognitive performance. And so knowing what direction these associations are in light of that is very informative. And then lastly, we only care about the brain, as interesting as it is because it's the what creates the output of our cognition. And so understanding cognition is very, very important. It's the end goal that sometimes we forget about because we are so enamored with all of our brain imaging techniques in studying the brain. And so while it's obviously very interesting and very fun to understand how the brain functions and how the brain ages in its own right, we need to not forget that we're studying it for the purposes of understanding cognition. So that's just kind of my three facets that I think people should keep in mind as they do or consume results from aging studies. So, okay, so I'm sure that I do not need to explain the fundamentals of how the brain ages to this group. So kind of just as a jumping off point, and here is a sagittal view of an MRI of a research participant from our study who's in their early 20s. Here is that same slice through the brain of one of our participants who is in their mid to late 70s. And just with the naked eye, you can younger person, older person's brain.
Kristen,
Yes.
sorry if I interrupt you. You might be touching your mic with your hands, maybe, so we hear like background noise.
Oh, sorry. Okay. I am wiggling my cursor to kind of show what I'm pointing at, but if you
This is
continue.
fine now, but like before we could hear.
Okay. All right. I will try to.
Sorry for interrupting you, but
No, please do. Thank you. I will try to not do that. Okay. Yes. So just looking at the difference between a younger individual's brain and an older individual's brain, there's macroscopic changes that are easily evident. You can see sulcal widening, you can see ventriculomegaly, but what these are is they're just endpoint non-specific markers of a myriad of biological processes that are happening at the cellular and molecular level. And so this is just the outcome. And so yes, the brain's macrostructure does change profoundly with age, but measuring it at that level, we really don't understand why or what the mechanisms are. And so as a part of this crash course of kind of brain aging, I wanted to go through and include what some of these most salient and profound changes are that we experience to the brain with age. And so, yes, the macrostructure changes with age, but what's under the hood? What is it that's driving this? And so some of the most profound changes that happen to the brain across the lifespan is that we have a substantial loss of certain, but not all, dopamine receptors as we age. And this is even relative to the loss of other neurotransmitters that we lose with age. Dopamine seems to be above many of those. And so this for sure is going to have some consequences on cognition. What we do know is that there's no frank neuronal loss with healthy aging. That only happens when we have some type of pathological neurodegenerative state. So we're not losing our neurons as we age in cognitively normal aging. But what is happening is there's fairly profound alterations to our neuronal processes or our neurites. And so we have quite a large reduction of the dendritic arbor. We have both a loss of and a shortening of the dendritic spines, especially on pyramidal neurons. And then even on the pyramidal neurons, there are large differences between the apical and the basal dendritic structures. And so there's large changes there. We have alterations to not only the myelin, but we have an actual alteration to the morphology of the myelin sheath. All kinds of odd things happen to our myelin sheath as we age. We have ballooning, either with inclusions or without inclusions. We have over loops of myelin, because as long as we live, we continue myelinating. And at some point that outpaces probably the amount of myelin that's optimal. And so we have this going on as we age. We have quite a bit of changes to the intracortical, intralaminar white matter alterations. This is really just being able to be studied in vivo, and it's very interesting. And this is in addition to the white matter and myelin alterations that we see deep in the subcortical white matter. We deposit heavy metals, increasingly so across the lifespan, of all sorts. Probably the most problematic of these is the deposition of iron, because then it sets off a reactive oxygen species cascade and all of the kind of neurotoxic things that can happen from that. Again, I probably don't have to tell this group that even in cognitively normal individuals, there's a deposition of beta amyloid plaques, and anywhere from 25 to 30% of cognitively normal older adults above the age of 60 or 65 or so carry the same amount of beta amyloid in their brains as someone with MCI or an early AD diagnosis, and yet they're cognitively normal. And so we have this to deal with as we age. Tau tangles exist in cognitively normal aging, but it's a very rapidly changing field. And so we're still as a field trying to figure out how much of that is in cognitively normal aging. And again, a lot of that work is done by people here at this center. And I see that Beth Mormino recently was here to give you a talk. And so I'm specifically not going to talk about much about beta amyloid or tau today, since you just heard a talk about that. We also have degradation to basic but important membranes throughout the brain. And so the blood-brain barrier becomes leaky with age. The boundaries between interstitial fluid compartments becomes weakened. These types of things happen. And there's several types of vascular risk-induced changes that seem to most preferentially affect the white matter. So all of these things and more are going on across the lifespan as we're aging. And so when we look at these macrostructural processes, outcomes in the brain, we have to keep in mind that it's an aggregate of all of these things that are happening to us biologically. And we have to remember that these are all biological processes, and the variables of interest that we would want to capture and measure are happening at the cellular and molecular levels. And so what we want to do is we want to study this and learn about this in living individuals, preferably in a repeated manner, following them across time. So these repeated in vivo studies require neuroimaging. And we have to keep in mind that these neuroimaging tools that we enjoy using are merely proxies for these biological processes that are happening over here. And so that's kind of a thing to just always keep in mind. And so keeping these things in mind, we have been running a study, and by we, I mean Dr. Karen Rodrigue and myself. We've been running a study that we started this in 2012. So as kind of a backstory, we each had K99R00 awards simultaneously, realized that we had some different neuroimaging interests and some common neuroimaging interests, and why put two samples of participants through all of these MRI scans and all of these cognitive batteries. And so we joined these projects together and collected during the R00 phase this cross-sectional lifespan study of about 208 individuals, and then realized that this would be a cheap, easy way for NIH to get a longitudinal study, because we could do a 5-year R01 where wave 1 was already collected for free from a previous mechanism. And so that's why how we've been able to get 8 years of longitudinal change done in a 5-year R01. And so that's what we're working on right now, just to kind of give you the backstory of our data set. And so we have a group of individuals age 20 to, our oldest person right now was 98 at wave 2. I'm not sure where she is at wave 3, but basically the participants come in for a comprehensive cognitive and neuropsych battery. And they had certain candidate genes, single nucleotide polymorphisms collected at wave 1. They had this MRI session that I'll talk about in a minute. And then they had a PET visit for beta amyloid PET ligand scanning. Came back 4 years later, started this R01. We redid the cognitive and neuropsych battery in the MRI, but not the PET. And then now we are currently, I think on our 3rd participant is in the scanner right now, actually, starting our data collection for wave 3. And luckily for us, this was the pandemic, and we just happened to luck out that we only had 2 or 3 subjects left at wave 2 when the pandemic shut everything down. And we've just now is time to come back for the 4-year lag, and we're actually able to get back to research again. And so this was very, very fortuitous for us that didn't wreck our longitudinal study, as I know it has for a lot of people. So yes, they have 2 visits to the lab, about 2.5 hours each, and they get multiple measures of all of the important domains of cognition for cognitive aging. So they have measures of processing speed, both crystallized and fluid intelligence, many different aspects of episodic memory, working memory, many different types of executive functions, as well as a booklet of psychosocial questionnaires that they fill out. And then we have not only a very strict screening for health conditions, because we kind of want to get at kind of best case scenario picture of healthy aging, but we also catalog and use as covariates when we can different types of health factors, such as different levels of vascular risk. And so as proxies of all of these things that we're interested in, from all these biological processes that are happening to us as we age from the last slide, we have a wide set of imaging sequences that we give in the scanner. Obviously, I'm not going to present all of these data to you today. I don't want to bore you with that much content, but just so that you kind of have an idea of what the participants are going through and the information that we have 2 task-based Fmri measures. And so we look at both mean bold and bold variability, as well as effective connectivity. We're very interested in iron deposition, especially Dr. Rodrigue, that's part of her expertise. And so we have a susceptibility weighted imaging sequence. We're interested in local regional perfusion. And so we have a PCASL sequence. We're also very interested in the volumes of the different hippocampal subfields. And so we have a high-res T2 proton density sequence for that. I already mentioned that we have a PET ligand. We use Amivid or florbetapir in our lab. We have measures of neurites by using multi-shell diffusion and biophysical models. And we have all of the classic measures of cortical ribbon, so volume, thickness, surface area, as well as volume of all of the subcortical nuclei. And then I've always been very, very interested in white matter. And for some reason in graduate school, that was the thing that excited me the most. And it's never faded. So I've always been preferentially interested in white matter. So I have many different ways that we try to get at properties of white matter. White matter compartment volume. We have different types of diffusion metrics. Fractional anisotropy probably is one of the things I'll talk about most today, because that's what we have published with previously using tractography methods. We have T1, T2 ratio sequences to look at myelin water fraction. We have FLAIR images to quantify white matter hyperintensity volume. And so as a package, we're trying to get at these major things that happen to us biologically as we age, with the hopes of using a multimodal imaging approach to see how these properties are related to each other, and then how these associations end up informing and predicting how we age cognitively. So that's the goal of the study in a nutshell. And so to come back to this schematic, there's really two foci to our study. And so you can think of them as, one, the genetic and health and lifestyle moderators of the effects of aging on brain structure and function. And then the other focus is on the interplay of brain structure and brain function, and how it leads to and predicts one's course of cognitive aging. And so this is a very interesting talk that I also can give to people, but today I decided I wanted to sit and focus just on the recent work we've done here. So everything I talked to you about today is going to be on these structure function associations, and then if I have enough time, maybe some of the structure association work that we've done. Okay, so I showed you at the very beginning of the talk that there's a wide range of variability in cognitive aging within the sample. And this heterogeneity in cognitive performance is likely yoked to the heterogeneity that we see in brain structure and brain function. And so to give you a little bit of a flavor of this, here are 5 kind of random examples of lifespan plots from some of our studies. And so here is one for volume. And so you can see that cross-sectionally, volume of the gray matter in the dorsolateral prefrontal cortex really takes quite a precipitous decline across the lifespan. But you can see for sure that there are individuals who have larger volumes, and there are individuals who have smaller volumes. And of course, this all comes down to needing longitudinal data and talking about intercepts and slopes. And so it could be that these older adults just started with a very, very high intercept when they were young and have maintained that. That's probably unlikely, but it's certainly possible. And so what we hope is that we end up with retained brain tissue, and even better, maintaining that brain tissue that we have, as opposed to losing that brain tissue over the years. And so here's an example from volume. Here's an example from thinning of the cortical ribbon. And so this happens to be the pars triangularis. And so again, you can see this is one of the regions of the brain that kind of has the hardest hit from the course of normal aging, but again, quite a spread of variance, especially in older age. And so there's individuals with thicker cortex in this gyrus versus individuals with much thinner cortex here. Here is a plot of white matter hyperintensity volume. And so we all hope that even in middle age, we're one of the people who has fewer of this white matter lesions. And especially when we're older, that we're, kind of free from this leukoariosis. But there are individuals who aren't in that category, right? So even in middle age, they kind of already start with this burden. Probably some type of small vessel disease is already brewing in this individual. And then in the older adults, there are individuals who have more. Same with white matter properties. So this is FA from probabilistic tractography. Here is this in this example from the cingulum bundle. And again, you can see that there's a wide variance in not only FA, but also in the trajectory of FA. So here are some individuals who are in their 80s who have a very high intercept in pretty shallow slopes. Here are some individuals that have already a low intercept, but then also maybe a steeper slope. And so all of these properties of brain structure, it's generally fairly known the direction and the nature of these properties. Generally, more tissue is better. Generally, higher FA and white matter metrics are better. But there's just as much variability, if not more sometimes, in brain function. And so here's just a plot of bold parameter estimates across the lifespan from participants age 20 to age 90 and beyond. And you can see that there are individuals well above and below this regression line. And so what's less clear is if you had to assign yourself to one of these circles, it's not as intuitively clear which of these is beneficial and which is detrimental. And so one thing that I want to talk to all of you today about as we talk through these Fmri studies is that obviously the bold signal doesn't have an interpretable intercept like many of these other types of metrics do, and to kind of highlight the importance of yoking these age effects on brain properties to a cognitive outcome variable to help with their interpretation, to see if maybe increased brain activation when you're old is a beneficial process, or maybe this is just older adults having to really ramp up their activation really hard, and maybe that's inefficient. And either of the two could be true down here, right? Maybe these are the older adults whose brains are aging well and they're highly efficient in their activation, or maybe these are older adults who aren't able to ramp up their brain activation as much as they should. And really the way to understand that and tease that apart is to look at how it predicts cognitive measures. And so we try to do that in most of our studies when we can. Okay, so just as one example of this and kind of a primer on what aging of brain function has looked like, we generally, the bulk of the literature has put groups of young adults and group of older adults into the scanner and then contrasted not only across the cognitive interest, cognitive measure of interest as a contrast, but then contrasted old versus young individuals. And what that has done is led people to refer to brain activation as either older adults, either overactivating or underactivating relative to a separate group of younger individuals. And so for lots of reasons, this isn't ideal. And because age across the lifespan is a continuous variable, bold estimates are a continuous variable, we tried to take the approach of doing regression-based analyses where we look at the effects of age or whatever properties we're interested in on the bold response at every voxel to try to see what those trajectories are across the lifespan. This is also helpful because it might be that those important transitions in whatever is altering in our brain function from when we're young to when we're old, those are likely happening in middle age. And so young, old, extreme group contrast can't really capture that. And so that's kind of an important thing that we try to put into our work too. And so here's just an example of a study that I wanted to show, because depending upon the way you look and the contrast that you're interested in, we can find either increases or decreases in brain activation. And so here's just a simple block design study that had two conditions. And so you had to make a living or non-living semantic judgment to blocks of nouns. And so some of these are very easy and straightforward. Cat, house, tree, those are very, very easy. The other ones by design were difficult. And the way to make it difficult was to make it ambiguous. And so a living or non-living judgment on these evoke some type of ambiguity. You have to process deeper. And so things like a sponge, a virus, a ghost, a speaker, these are things where you really have to kind of stop and think, is that living or non-living? And we hypothesize that they really should bring online extra parietal and frontal activation to handle these types of decisions. And so those were our contrasts of interest. And so we can look at just the effect of making these easy decisions. And you would think that that would probably bring online this kind of canonical semantic network of regions. And then we wanted to see what the lifespan age effect was on that process. And so what you can see here is that all of these clusters are on the warm end of the scale. And so that means that these are all regions of the brain that increase in their activation while making this easy semantic judgment as age increases. And so you can just think of this as the voxel-wise product of that age on bold regression. And so if you only looked at this, you would have the idea that, oh, brain activation increases with age. Okay, that's interesting. I wonder why that happens. And just to give you a visual of how that looks in some of these random regions, you can see these increasing trajectories of bold estimates as age increases. And so during this easy task, your brain is having to activate more to accomplish this. The older you are. When we change and then look at, to me, this probably more interesting contrast is what happens when you have to switch from doing something that's easy to doing something that's harder, and look at the effects of age on this demand-related bold modulation. And so that is what we looked at here. And what we found here, you'll see, is that all of these clusters here are on this cool scale, meaning that these are decreases with age. And so this is an age by demand interaction at every voxel. And so these are the regions where individuals significantly increased their activation from easy to hard conditions, and then where that kind of regression decreases with age. And so the older we get, the less we are able to modulate these regions to difficulty. And just to glance around what these clusters are, you can see that these are regions of kind of our cognitive control systems, frontal, parietal, striatum, cingulate types of regions. And again, just to kind of, and you'll note that there's no regions that increase in their modulation as we get older. And so just to kind of see what those look like, you can see these different plots here. So for example, the caudate, you can see this ability to modulate to difficulty decreases with age. So I showed you this because it's an example of, depending upon how you look and what your question is, you could get both answers to how does brain activation change with age? Does it decrease? Does it increase? Well, the answer is that it's both. And the way to tease apart which of these things are maybe beneficial or compensatory or are detrimental and a failure to kind of ramp up what is needed lies in yoking this and interpreting this in the context of their cognitive performance, whether it's accuracy or something from in the task while they're being scanned, or their cognitive performance measured from out in their cognitive neuropsych battery. And we tried to look at both of these things as we do these studies. So what I would like to spend the rest of the time today doing is walking you through a few of our recent studies where we've used this multimodal imaging approach to look at structure function associations and also structure associations, but I'm pretty sure that I'll end up skipping these studies to give a little more time to talk through these. And this is just as an example using variables such as beta amyloid SUVR from PET, bold variability and mean bold from our Fmri studies, diffusion metrics from tractography, cortical thickness, hippocampal subfield volumes, iron accumulation, these types of things to try to get at some synergetic associations among different brain properties. So I'll start out with this study that we did. This was part of the requirements for the dissertation for Jenny Reek, who did a lot of this work. And so there is a well-known phenomenon in aging and looking at brain activation called dedifferentiation that a lot of you are probably familiar with. And so we were interested in whether knowing kind of the quality of the white matter connections that innervate, if you will, the different gray matter regions that are activating to make these types of perceptual contributions, whether that can be predicted from knowing kind of the white matter quality of that. And so we did this study where we isolated a selectivity measure of activation for the fusiform face area. And so we have a selectivity metric for how selective the voxels are to activating to faces in the fusiform face area. And this really declines with age. And this replicates what the whole literature has found. So this is dedifferentiation. We also looked at this in other parts of the ventral visual cortex and making more broader types of selectivity. And so this was for whether or not the stimuli were animate or inanimate. And this selectivity metric also declines with age. And so you can see this here. You can see just the general greater activation of this fusiform face area when in the presence of viewing faces greater than viewing houses. And then you can see that there's this differential response to this more medial region here being more selected to inanimate objects, and this more lateral region here being more selective to animate objects. And then you can see that these decline in their selectivity with age. And so we have both selective and specifically selective, and then broader selective metrics. And so we were interested in whether the white matter that should run through and connect most of this, which would be the inferior longitudinal fasciculus, we wanted to know if each individual's metrics from their ILF tracts would have any predictive power for this preservation or decline in selectivity or dedifferentiation. And so we did probabilistic tractography to isolate the ILF in every participant. And just like all other association tracts in the brain, how we would expect, we see a pretty steep decline in this case of FA in this tract. So this is negative 0.57. And so then we wanted to know whether this FA along this tract had any predictive power for either of these two metrics. And what was interesting was that using mean FA from this entire ILF tract did not significantly predict just selectivity in general. And so instead, what we found is that if you only look at the portion of the ILF that contains the fusiform face area, so this middle segment of the ILF FA, you can see that as you kind of have higher FA in this tract, the more selective you are. And so less de-differentiation, if you will, in the individuals who've maintained kind of better white matter quality. And so this was the specific association. And then we found that when it came to this kind of broader object properties, so whether something was animate or inanimate, it was actually the whole length of the entire ILF tract that significantly predicted this ventral visual cortex selectivity. And so we thought this was really interesting because this seems like this kind of fundamental perceptual sensory type of process that's happening with aging, where you kind of fan your variance and you become less and less specific as you get older. And we found that in part, this is because the white matter that's kind of running through these regions, its quality is not as good. And this was a passive viewing task. And so in our first structure function study, we weren't able to yoke this to cognition. And so this spurred us to go on to look at tasks that were a little more cognitive in nature. And so the next study I want to show you is where we did just that. And so we looked at the associations among aging, white matter metrics, Fmri modulation to difficulty, and then its association to cognitive performance. And this was done with a postdoc in the lab, Dr. Christina Webb. And so we have an in-back task that we give to all of our participants in the study. And it's it has a very wide range of difficulty. So we have a 0-back condition, but then we also have a 2-back, a 3-back, and a 4-back condition. Everyone practices this in the mock scanner before they go down the hall and get in the real scanner. And people all hit about 80% accuracy or so on these trials. And so one thing that people are sometimes astonished with is that the older adults in the study can do these more difficult conditions, and they for sure can, which is also nice for us, knowing that as we age, hopefully we'll turn out that way too. So what we do is, because we have this linear parametric difficulty contrast that shows what happens when we increase activation to working memory load, we have these outcomes. And so there are regions of the brain that increase their activation to this increasing linear parametric load. And you can see those in the warm colors, and we refer to those as positive modulation. And then of course, there's regions in the brain that decrease in their modulation simultaneously as difficulty increases. And we call that negative modulation. And so both positive modulation and negative modulation are reduced with age. And so just think of being farther from 0 as better, right? And returning back toward 0, bold modulation would be worse, right? And so we find decreases in both of these. And so then in this first study, we thought we wanted to kind of cast a wide net and have kind of a representative search for structure function associations. And so what we did is we took 6 white matter tracts from the brain. We got these from deterministic tractography this time. And these are all regions that are known to have some type of association with higher order cognition, cognitive control, but also pass in some way through the prefrontal cortex. And so we have the cingulum bundle, the inferior frontal occipital fasciculus, the uncinate fasciculus, the superior longitudinal fasciculus, the inferior longitudinal fasciculus, and the genu of the corpus callosum. And because this is a complicated multivariate question that we're trying to ask, we decided the best thing to do in service of answering this question would be to specify a structural equation modeling with the hypothesis flow of variance that we think captures this question that we have up here. And so that looks like this. And of course, I don't expect you to kind of take this all in, but I'll tell you the gist of this. And so basically we have age predicting FA as a latent construct, and then the observed indicators of this latent construct is FA from these tracts. And then we made a latent construct of both positive modulation and negative modulation. And the clusters that you see over here in red are the ones that are observed indicators of this latent construct. And then the same for these clusters in blue over here are the indicators of this negative modulation construct. And then we wanted to know is if these, one, were there structure function associations here that were meaningful, and two, if then they had any kind of cognitive consequences. In other words, so what? If you find brain associations with age, it kind of only matters if then it predicts cognition. And so we have, as a latent construct, accuracy from the in-back task in the scanner. And so those observed indicators are 0, 2, 3, and 4 back. And then we have an executive function construct that's made from the higher, harder switching indices of the variables on the DCAF's measure of executive function. And so we have those there. And then so our primary hypothesis of mediation, what we would hope to find would be that the age effects on these cognitive measures are mediated by knowing kind of the white matter quality of these tracts, and if this white matter quality has anything to do with bold modulation, and if that mediates the association with cognition. And so you can think of these triangles. So age affects white matter, affects modulation, which then affects two different measures of cognition, and then the same thing through negative modulation. And interestingly, what we found is we did find that we have partial mediation here of the fact that age effects on positive bold modulation is mediated by one's FA. And so this brings us down to non-significance, not that there's any reason in the world for me to try to convince you that white matter completely mediates and accounts for the age differences in activation. For sure, we don't think that's true, but statistically, that's true here. And then that this indirect effect, this mediation, significantly predicts performance on not only performance while you're in the scanner doing the task, but also on out of the scanner cognitive performance. And so this mediation was very, very strong. Interestingly, this isn't the case for these kind of deactivating negative modulation regions. And so the age-related reductions in FA did not significantly predict negative modulation. Of course, we found the expected association where there was a reduction of modulation with age, and reduced negative modulation did predict poor executive function and poor in-back accuracy. And so this was interesting, and I can tell you later, if you're interested, my hunch of why it's positive modulation has a strong mediation and negative modulation doesn't. And so we were very happy with this because we feel like we've understood something and yoked this to real cognitive measures now at this point. And then we realized that this was kind of a catch-all set of white matter connections. And this is interesting, and this gave us this overall picture, but this postdoc and I started wondering if we could zoom way down on this and see if we could find specific effects of specific white matter tracts on modulation in specific regions. And so that inspired us to do this next study. And so in this study, we are interested in the associations among aging of specifically the frontostriatal tracts, white matter properties, and their effects on fMRI modulation, and then what cognitive correlates there were of that. And so as we started looking at the literature, we were kind of surprised that there isn't really kind of an out of the box frontal striatal tract. And so we were very happy to work to get a nice tract. This, again, we used deterministic tractography here. And what we did is we found and isolated these tracts for every individual, basically seated in more dorsal striatum, and then connecting to more dorsal, medial, and lateral prefrontal cortex. And that obviously was just because we weren't as interested in those types of fibers that came out of the ventral striatum and then went to like orbital frontal cortex for obvious reasons. And so we were very happy with this tract. And because we hadn't ever worked with this before, we didn't really see this in the literature, we kind of wanted, to make sure that this was behaving in a way that we expected. And so we know that this frontal striatal circuitry in the brain biologically really takes one of the biggest hits of all the white matter in the brain during aging. We know that from primate and human research. And so we also found quite a steep slope of reduced FA with increasing age. And then we also looked at mean diffusivity, and we find what we find in others across the world all find when we look across the lifespan. We find quadratic accelerating increases in diffusivity with age. And so we found a very steep slope of that too. And then again, kind of for some real world face validity reasons, we wanted to kind of make sure that this predicted higher order cognition in a way that you expect it should. And so we did find a significant interaction where age significantly moderated the effect of frontal striatal FA and MD on our executive function composite from our cognitive neuropsych battery. So frontal striatal circuitry really is affected strongly by age, and it has a negative effect on executive function performance. And so then we wanted to know, then, does FA or MD in this tract, not only does it affect bold modulation to difficulty, if so, where? And so we're of the very strong opinion that when you look at these types of associations, the better way to do this is take the variables that you're interested in and directly model them voxel-wise into your whatever you use, SPM, in our case, search. And so that's what we try to do in all of our studies like this. And so what we did is we entered frontal striatal FA, and then in a different model, frontal striatal MD, and age and their interaction into the voxel-wise GLM in SPM. And what this does is it allows us to ask the question, where in the brain are the voxels that are significantly associated with one's FA from this frontal striatal tract? So this is a whole brain voxel-wise search. And so what we found is that poorer age-related frontal striatal MD was significantly associated with lower bold modulation as working memory load increased. And we found this selective to clusters in the striatum, in the inferior frontal gyrus, and into the insula. And so we thought this was really interesting and surprisingly specific. This could have been many different places in the brain that this turned out. And so because we kind of wanted to make sure that this was a robust finding, we went ahead and ran this exact same thing where we took these frontal striatal tracts and we used them to predict bold modulation on our other fMRI task that everyone in our study gets. And it's just a spatial distance judgment task. And it also has a parametric modulation of difficulty. And so we found almost the exact same thing in that task. And so that's why you see two colors here. And so these pink colors are the significant clusters for the NBAC task. And then these blue clusters are those significant regions for the distance judgment task. So I thought this was really interesting and really specific. And before you probably have already started to think, wow, that's also an interesting hemispheric dissociation. We kind of thought that too, but we're hesitant to really go hard in that direction of interpretation because, you know, thresholding in Fmri is kind of a fickle practice anyway. And so if you loosen this strict threshold up a little bit, all of these clusters become bilateral for both tasks. But yes, at this strict threshold, it does track with the fact that in back by design, we wanted a verbal and a nonverbal difficulty task in our study. And so you can see that the left and the right go with these two tasks, but we didn't really interpret that. And then lastly, what we thought was really important, of course, was to yoke this to cognition. And so we did find that this age-related white matter tract degradation of the frontal striatal tracts related modulation reduction. So that's the structure function association together in a phrase, was associated with poorer in-back accuracy during the scan task, which you see here. And then also on the executive function, which I didn't put here just for space. And so the older you are, the poorer your frontal striatal FA is, the less you are able to up modulate activation, fold activation to difficulty, and is predictive of poorer performance both during the task and on executive functions in general. And so this, again, we were happy with this kind of being able to complete the whole triangle of aging, brain associations, and then cognitive performance, which we think is helping us understand a little bit of how things are working together in the brain to try to put together what we know about cognitive outcomes. And so I'm looking at my watch. And so I think from here, I just want to go to my conclusion slide. If I can do that in a non-clunky way, I don't think I can. Whoops. Yeah. Sorry about that. So this is a line of research that we're excited about. There's obviously many directions that we plan to take this and have already taken this that I couldn't talk to you about today, given all the different neuroimaging properties that we're able to measure. And so that's what we're continuing to work on. And so I'd just like to conclude by saying that, yes, the aging brain is for sure characterized by many different types of alterations, mainly losses to gray and white matter structure, but also alterations to the bold response. However, this really is predicted and driven, I think, by the individual differences in our health and lifestyle factors that drive our ultimate brain properties and work together with our brain function to kind of determine what our cognitive outcome is. The reason we can find these associations is because we use this multimodal approach so that we can kind of see how things age in tandem rather than independently. And then again, we think that yoking these associations to cognition really helps us kind of understand the nature of the changes in these brain properties. And then most importantly, probably for me to say, is that everything I've shown you today almost has the major caveat that these are just snapshots in time of individuals in a cohort. And so these cross-sectional findings are an interesting stepping off point, but the only way to understand real aging is to see how individuals with all of their own individual differences kept with them over time in a longitudinal study is the only way that you can really talk about lead lag associations. And so all of these cross-sectional studies are kind of to be determined to see how they turn out with the longitudinal follow-up data. So thank you very much for your time. Thanks to all the great people in the lab who did so much of this work, and thanks to our funding agencies for allowing us to be able to do this work at all.
Thank you very much, Kristen, for the nice talk. So we are a little bit late, but we do have time for questions. So if people have questions, please address them. Meanwhile, I can start with a few. So yeah, the first one, I think you just addressed it, but it was more of a clarification than a real question. I think you showed at the beginning you are acquiring longitudinal data. Okay, but all the results you presented were on cross-sectional data. So yeah. Okay, so and you're planning to then analyze that data.
Yeah, so what we've done during the pandemic was sit and sift through and pre-process and process and work through a lot of the change data. And the more we get into this, the more we very quickly realize that there's not a ton of cross-sectional imaging studies at the level at some of these kind of newer types of variables. And so a surprising amount of work during the pandemic has been kind of figuring out ways to normalize things across waves. Luckily, we haven't had any scanner changes across 3 waves, and we haven't had any major software updates, but we've realized to do this really well, there's a lot of probably signal to noise modeling and just, comparing phantoms that we scanned at each time before we really go into the data analysis, because it occurred to me that if you find maybe change in the direction you don't expect in your results, you then don't want to go back to pre-processing because that seems biased and unfair. And so we've decided to thoroughly go through all the QCing and just kind of see any differences in shift or drift in the scanner over time, how to regress that out of the data before we send it to kind of like second level like group data analysis. So that's, and we've just finished that up, and we've started our first submissions of abstracts to conferences on our actual longitudinal findings, 2-wave. And so we've just started collecting wave 3, I think last week.
Okay, thank you. And I guess another question I had, I'm always very skeptical about, FA.
Yeah.
Because it can be confounded by so many things, including crossing fibers or partial volume. So I don't know, I'm just curious to know how confident you feel in, in this measure, and whether you are planning on supporting these results by looking at other more specific white matter measures.
Yeah. That's a great question. So at wave 1, we had a diffusion tensor sequence. And so the cross-sectional stuff used that. At wave 2, we were able to add these multi-shell diffusion measures, and we're in the process of analyzing that too. And so we'll have longitudinal change in that in wave 3. And so what we're starting to do now is trying to run some of these same analyses with our kind of NOTI metrics to see if we get the same findings as we did using the tensor-based things. But I'm completely in agreement with you. FA would never be my metric of choice. We do a combination of probabilistic and deterministic tractography on all the tracks we're interested in, knowing that each has their strengths. One kind of deals with, kissing crossing fibers a little better. One's more conservative, and so we kind of do them both and get FA out of each of them, and MD and RD, AD, whatever. And we do a lot of in-house kind of like harmonization to kind of see how these tracks kind of should perform before we move them forward to actually use them in analyses. But yeah, I'll be very happy to get away from FA and start getting to look at some of these better better indices.
All right, well, looking forward to those results then. I don't see any other question coming up, so I think I'm gonna wrap it up for today. So thank you very much again for presenting, and thank you everyone for attending today.
Yeah, thank you very much.
Have a good day. Bye.
Bye-bye.
Bye-bye.